Namaste to all the students. Khushbu myself JV'n Dr. Khushbu Verma, Faculty of Agriculture and Veterinary Science. Today we will discuss about meiosis first stage, prophase first. Prophase first has major events in meiosis 1, and basically it is responsible for all kind of crossing over and recombination. So first of all we will know about the history of meiosis. So meiosis was firstly discovered and described by in the sea urchin eggs in 1876 by German botanist Oscar Hartwig. It was declared again in 1883 at the level of chromosomes by the Belgian zoologist Edward van Beneden in Ascaris worm's egg. The term meiosis was coined by J.V. Farmer and J.V. Murray in 1905. Where it occurs, so meiosis occurs in eukaryotic life cycles involving sexual reproduction. It always occurs in only reproductive cells. In lower plants, meiosis occurs after fertilization in zygotes. In higher plants, it occurs before fertilization in the time of gamete creation or gamete formation. Facts about the meiosis. So meiosis is a type of cell division by which germ cells are produced. There are two types of meiotic divisions for meiosis 1, which is true meiosis, and meiosis 2. And it's called original cells are diploid in nature, but after the completion of meiosis, 4 daughter cells will be produced and they will be monoploid. Basically, if we talk about the definition of meiosis, so daughter cells will have half number of chromosomes after the completion of meiosis process in comparison to their parent cell. If parental cell have 4 chromosomes, so daughter cell will have only 2 chromosomes. Daughter cells, as I told you, daughter cells contain half the number of chromosomes as the original cells, and by the process of meiosis, gametes production has been performed, like egg and sperm cell will be produced, occurs in testes in males and occurs in ovaries in females. During meiosis, DNA replicates once, but the nucleus divides twice. Meiosis 1, the reduction division, diploid mother cells produce 2 haploid cells. So that's why we call it true meiosis process, where the two daughter cells will be produced after the meiosis 1 completion, and these daughter cells will have half number of chromosome in comparison to their original mother cell. And this meiosis 1 have 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 again subdivided into 5 stages, sub-stages. First is leptotin, second zygotin, third is pachytin, fourth is diplotin, and fifth is dikinesis. All the crossing over and recombination process, chiasmata formation, all these creatures, all these creations will be done in prophase 1. So that's why today we will discuss only on prophase 1 stage. During prophase first, DNA is exchanged between homologous chromosomes in a process called homologous recombination. This often results in chromosomal crossing over. The paired and replicated chromosomes are called bivalent or tetrads. The process of pairing the homologous chromosomes is called synapses, and at this stage non-sister chromatids may cross over at point called chiasmata. First of all, we will discuss its first stage, which is leptotin, and leptotin is also known as leptonema from Greek word meaning thin threads. During this stage, individual chromosomes begin to condense into long strands within the nucleus. However, the two sister chromatids are still so tightly bound that they are distinguishable from each other. And in zygote, next stage is zygotin, and in the zygotin stage, it is also known as zygonema from the Greek word meaning paired threads. Zygotin occurs as the chromosome approximately line up with each other into homologous chromosomes. The combined homologous chromosomes are said to be bivalent in this process. Next one is pachytin. In pachytin or pachynema, the homologous chromosomes become much more closely associated. This process is known as synapsis, and the synapsed homologous pairs of chromosomes is called as tetrad because it consists of 4 chromatids. It can't be observed until the next stage, but the synapsed chromosomes may undergo crossing over in pachytene stage. The chromosome continue to condense. Next one is diplotin. During the diplotin stage, also known as diplonema, from Greek word meaning two threads, the homologous chromosomes separate from one another a little. Chromosomes themselves uncoil a bit, allowing some transcription of DNA. Next one is dikinesis process. The chromosomes condense further during the dikinesis stage, from Greek word meaning moving to. This is the first point in the meiosis where the 4 parts of tetrads are actually visible. In this stage, homologous chromosomes separate further and the chiasmata terminalized, making chiasmata clearly visible. Here we can see that 2 chromosomes are just attached at their telomere region, and that's why this structure has been created, which is known as chiasmata. This is the flow diagram where we can find out the different stages of meiotic prophase 1. As I told you, first is lactotin, where chromosomes will start to condense and they will be separated. Here no bivalents are arranged. And next is jagotin, where bivalents will be generated, will be arranged. And next one is pachytene, where tetrads are formed, or you can say synapses will occur. In diplotin, synapses will be deformed and 2 chromosomes will move apart from each other. And last one is dikinesis, where chiasmata will be more visible, but chiasmata will start to form in diplotin stage. Main characteristic of prophase 1 is crossing over. During prophase 1 of meiosis, a part of chromatid of one chromosome goes over to a chromatid of other chromosome and vice versa. The interchange of the part of chromatids of a pair of homologous chromosomes is known as crossing over. Crossing over is a process by which 2 chromosomes of a homologous pair exchange equal segments with each other. Crossing over occurs in the first division of the meiosis. Here we can see means it will be occur in meiosis 1, means prophase 1 stage. In the prophase 1 stage it will be occur. Here we can see that this is homologous chromosome. They will make a synapsis and then they will exchange their region from each other. They will interexchange. So this is the phenomenon of crossing over. Here we can see that the 2 homologous chromosomes will make a pairing and will create the synapses, and because of the synapses, crossing over will be done, and then sister chromatids of one chromosome will exchange their part from another sister chromatid of another chromosome, and that's how crossing over will be done. What is the importance of crossing over? Crossing over takes place between the non-sister chromatids of homologous chromosomes. Crossing over is one of the most important events in meiosis because it allows variation in the produced offspring, produces new individual having new combination of traits, helps in establishing the concept of linear arrangement of genes, helps in mapping of chromosomes and selection of useful recombination. So this is the sum up of this session of Prophase 1, and this session is powered by digital version 2.0 JAYOTI VIDYAPEETH Women's University. Hope you are satisfied with this session, and if you have any query, then mention in the comment box. I will resolve it. Thank you.